Okay. Now, what to study in the chapter of molecular biology? Listen, every one of you, very correctly. In the molecular biology, in a single word, the entire lesson is exactly summarized in this particular step. Remember, we have to study the replication in prokaryotes and eukaryotes, transcription in prokaryotes and eukaryotes, followed by translation in prokaryotes and eukaryotes. All these processes we have to read very briefly and elaboratively. Now let's start the chapter very elaboratively. Okay, so listen to me for 20 to 30 minutes. After that, immediately we will start, we will discuss the doubts. Okay, you all people commonly know that, friends, who discovered the double structure of the DNA, double helical structure of the DNA. You know that James Watson and Francis Crick. You all people also know very well, before the James Watson and Francis Crick, Paul is also one of the leading scientists at that particular time. So in the year of 1953, Paul actually proposed what structure, friends? Before the Watson and Crick, actually, Paul proposed triple helical structure. Unfortunately, the triple helical structure of the DNA was not accepted by most of the scientists. So, friends, after Paul, the James Watson and Francis Crick, they proposed double helical structure of the DNA. Based on what? That's also a very important point. Based on the X-ray crystallography structure taken by the Rosalind Wilkins and James Franklin. So later the Nobel Prize was shared among 4 people actually. So based on the X-ray crystallographic structure, Watson and Crick proposed that the DNA is the double helical structure. See, friends, once they proposed the double helical structure, they have published their entire research in the world famous journal that is called Nature. Remember, these are all the last lines in the publication. In the publication, these are all the last lines written by the Crick. You know what they have written? See here, and this is the original diagram drawn by the Crick. Look into this, look into this. What is the last line they have written? Very interesting line. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. What they are trying to say here, it is not escaped our notice, and it has given an idea to us. How it has given an idea? The base pairing has given an idea. You know very well that adenine always pair with the thymine, guanine is always pair with the cytosine. So what he is trying to explain, so whatever the base pairing present in the DNA, it has given them an idea. What idea it has given them? That we have to propose a model. Why we have to propose a model? By using that particular model, friends, the DNA undergoing the process of replication. What the DNA is undergoing? The DNA is undergoing the process of the replication. That is what they understood. Anyway, see, all the students listen to me correctly. In a single slide, the entire lesson is this. So we are going to summarize the entire lesson in this particular slide. Listen to me very correctly. If I ask all of you a simple question, a very simple question, what is the function of the DNA? What kind of answer you people will give me? Many people will say that DNA is a genetic material, right? This is what people will say, that what is the function of the DNA. Now, all of you try to understand me, friends. The ultimate function of the DNA, whatever you said is right. The DNA is involved in the replication. Most unique function of the DNA is what, friends? Synthesis of a protein. Yes or no? Of course, yes. Now, let me tell you one very interesting point. All of you listen to me very carefully. In our body, how many number of cells are present, friends? You know, right? An average in our body, 37.2 trillion cells are present in the body. Friends, 37.2 trillion cells present in the body. Every function performed by the living cell is under the control of protein. Every function, what not? Let's take an example. If we oxygen can be supplied in our body continuously by hemoglobin, is it? What is hemoglobin? Which is a protein. Correct, na? Suppose example, an antigen enter in our body. What our body starts releasing? Antibody. Body starts synthesizing antibody. What is antibody? Protein. Can you imagine any body reactions without enzymes? No, you can't imagine. What are enzymes? Chemically proteins. Correct, na? So what not? Every function performed by the living cell is under the control of the protein only. Friends, that particular protein can be synthesized by the DNA molecule. That's the reason only we use it to say DNA is the most abundant molecule, not to get confused with that. Now, let me ask you one very simple and very interesting question. As I am continuously saying that the ultimate function of the DNA molecule is synthesizing the protein. Is it? Of course, yes. Now, all of you, please try to answer my question. If the DNA having the ability or capability to synthesize the protein directly, why can't it is involved directly in the synthesis of protein? Instead of why the DNA is synthesizing an intermediate molecule RNA? What is the reason? Can any one of you please answer my question? Why the DNA molecule is not synthesizing the protein directly? Any one of you please type the answer. Good. The simple reason is that DNA is extremely stable molecule, very, very highly stable molecule. So what I'm trying to explain to all of you friends, here RNA is involved in the synthesis of protein. Suppose example, RNA got damaged. RNA got damaged, then there is a no problem because we have the DNA. Now the DNA is going to give rise to the RNA. Then RNA can give rise to the synthesis of protein. But if the DNA got damaged, the same wrong genetic information will pass us to the next generation. As a result, what happened? Enter all the generations, it leads to the mutations. So what I'm trying to explain you, DNA molecule, in order to protect its stability, all of you try to understand me. DNA molecule, in order to protect its stability, in order to protect its integrity, it never involved in the synthesis of protein directly. Instead of involving in the synthesis of protein directly, the DNA is going to synthesize an intermediate molecule, that is the RNA molecule. Finally, RNA molecule will synthesize the protein. That's not a problem. Now all the students try to understand me very correctly without confusion. This is also a very important point. The DNA molecule, whenever it is synthesizing before synthesizing the RNA, first DNA undergo the process of the replication or duplication or cloning. You all people commonly know that we all look like our mother and father. Why? Because the genetic information, that is DNA, passed from their generation to our generation, which means friends, before synthesizing the RNA, DNA molecule undergo the process of replication, which is the cloning itself. After cloning is over, all the students try to understand me. Now the DNA is going to give rise to the intermediate molecule. What was that intermediate molecule, friends? Which we call it as the RNA, specifically mRNA. Now listen my words correctly. How can I give the definition of the RNA? Tell me, synthesis of RNA. Listen, DNA mediated RNA synthesis, we generally called as transcription. Now I want you to answer a very simple question. See, if the RNA want to be synthesized, do you think we require the entire DNA? Suppose this is the entire DNA. If we want to synthesize the RNA, do we require the entire DNA? What are friends? Do we require only a little part of the DNA? All the students remember without fail, if we want to synthesize the DNA, friends, RNA, we do not require the entire DNA. What is the use of the entire DNA? Of course, that is true. We require one strand of the DNA that is acting as a template. That is fine. In order to synthesize the RNA, we require only a part of the DNA. Now you have to ask me a very good question. Sir, DNA is a very, very big molecule, very big molecule. Now you have to ask me, sir, which part of the DNA that I have to take? Because it is a very big part. Which part I have to take? That's a very good question. It depends on what protein it has to be synthesized. Only that part we will take. Suppose example, friends, this is the DNA example. Now the function of this DNA is synthesizing a protein called as a collagen. It has to synthesize the collagen. So if it has to synthesize the collagen, suppose this portion of the DNA code for the information of the collagen, then only, friends, this information only pass to the RNA, not the entire DNA. So what I'm trying to explain you, during the process of the transcription, a part of the DNA can be transferred to the RNA. So that's why we can call the DNA mediated RNA synthesis, which we called it as, friends, a transcription, not to get confused with that particular point. Okay, the RNA got synthesized. Now, friends, RNA is going to synthesize what? Protein. So now mRNA contain information. What it is doing, with the help of amino acid, with the help of a ribosome, it is going to synthesize and it is going to fulfill the ultimate goal of the DNA. So what we can call the RNA mediated protein synthesis, we can call translation. Sometimes, friends, instead of calling RNA mediated protein synthesis, friends, you can also call it as, listen every one of you very correctly, you can also call it as a DNA guided. We can call this, you can call DNA guided RNA mediated protein synthesis, which we can call translation. Friends, all of you remember one point very clearly without confusion. Everything is under the control of DNA. Everything. It is, nothing is under the control of RNA. Always remember that. Whether the RNA want to be synthesized, whether the protein want to be synthesized, that is controlled by the DNA. It is not at all controlled by the RNA. Not to get confused with that. Now, all the students try to understand my point. Since many are, especially viruses, many organisms generally having DNA as their genetic material, we all people commonly know that. But friends, if you look into the RNA, viruses are having what, friends? RNA as their major genetic material. DNA is not their major genetic material. But every one of you know very well how the virus can show their action. Suppose you take an example of a HIV virus. When the HIV virus enter in the body, what happened? First, it release the RNA. From the RNA, it is going to synthesize the DNA, and that DNA is undergoing the duplication, and it is increasing the multiple copies. So what I'm trying to explain you, virus having the RNA as their major genetic material. Now, friends, so virus having the ability or capability to synthesize the DNA molecule from the RNA. So in this particular step we have studied, we synthesized RNA, is it? We synthesized the RNA from the DNA. So friends, synthesis of RNA from the DNA. What do we call it, this particular process? We call it this particular process as the transcription. But what we are doing, we are synthesizing the DNA from the RNA. So, which is a reverse process. That's why we call it as what? Reverse transcription. And what kind of enzyme is going to catalyze this particular reaction, friends? This reaction can be catalyzed by an enzyme called as a reverse transcriptase enzyme. Not to get confused with this particular point. I want you to understand it very correctly without confusion. Okay, now very important point. So here information is passing from where to where? From the DNA to the protein level, is it? So listen everyone, the mechanism, the mechanism or the process by which the information is passing from the DNA to the protein. This entire mechanism only, friends, we people generally call it as the central dogma. Not to get confused with that. The name central dogma was given by the genius scientist Crick. Friends, which is more important for the exam now? So the more important for the exam is remembering where all these events are taking place. See, remember, now my goal is once I complete the lesson, let's solve first the 2023 question paper, which was happened few days ago. See, in that there are 4 to 5 part C questions are there, 3 to 4 part B questions are there. Among this, you should be in a position to answer at least half of this, which indicates that your preparation and your level of understanding is absolutely right. See, for the CSIR people, we get nearly 20 to 26 marks we will get from this chapter. For IIT JAM people, they'll get nearly 3 to 5 marks from biotechnology chapter only. For GATE people, it is more than 10 to 14 marks you can expect from the molecular biology chapter only. So that's why we should not leave any topic or any concepts here. Okay, now the most important point is, friends, knowing the locations. Every one of you remember my words correctly, friends. In the competitive exams, 90% of the time, the questions will be always asked from where that particular event is taking place, where is the exact location. So that's the, see, in competitive exam you have to remember where, what, why, how. Then if you apply these 4 concepts towards any concept, all the concepts will become super easy to all of us. Now let's talk about it. Now you only has to answer all my questions. Let's take a prokaryotes, let's take a eukaryotes. In the prokaryotic organism, where the process of DNA replication will be taking place? In the prokaryotes, that process of DNA replication will take place where? In the cytoplasm. What about the eukaryotes, friends? In the eukaryotes, the process of DNA replication will take place where? In the nucleus. Right. Now, every one of you very quickly answer, in case of a prokaryotes, where the process of transcription will take place, that is the synthesis of RNA, that is in the cytoplasm. Where the process of transcription will take place in the eukaryotes, that is also in the nucleus. Every one of you very quickly answer my question. Where is the site of translation in prokaryotes and eukaryotes both? Where is the site of the translation both in the prokaryotes and eukaryotic organism? All the students remember all the time, it doesn't matter whether it is the prokaryotic organism or whether it is the eukaryotic organism. No, 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 no. That is wrong. ER, that is different, friends. In the cell, there are two areas protein synthesis will occur. That is on the ER membrane, because ER contain membrane-bound ribosomes. And other protein synthesis occur in the cytosol. This is the major protein synthesis. Okay, so now where the translation occur in the prokaryotes? In the cytoplasm. What about the eukaryotes, friends? Undoubtedly, in the eukaryotic organism also the process of translation is taking place in the cytoplasm only. Why? What's the reason? The reason is very simple, friends. All the translation factors which are required for the protein synthesis, that is exclusively present in the cytosol only. That's why the translation exclusively takes place in the cytosol only. Now, let me explain you the most important step right now. All of you listen to me correctly. Friends, if you take an example like a prokaryotic organism, let's take a bacteria. As just now we studied that in the bacteria, the DNA undergo the replication in the cytosol. DNA is going to give rise to the RNA, where in the cytosol only, and the RNA is going to give rise to what, friends? Give rise to the protein, where of course in the cytoplasm only. So DNA, then the DNA is going to give rise to the RNA. RNA is going to synthesize the protein in the cytoplasm only. All the students listen my statements not to get confused. I am talking about only prokaryotes. In the prokaryotic organism, the process of transcription and the process of translation, what do we call it as? We called they are coupled with each other. Such an important and interesting statement it is. What do you mean by that? Which means, friends, whenever a little part of the RNA comes out, all the students try to understand me correctly. Whenever a little, very little part of the RNA comes out, enough, immediately what happen? The ribosomes will come and attaches and start the synthesis of protein immediately. But do you think it will happen in the eukaryotes? No, friends, we are very complex organism. Our genome is a super complex when compared to the prokaryotic organism. So what I want you to remember, listen to me, this is the DNA. Where the DNA replication is happening, you all people know that it happens within the nucleus. Once the DNA replication is over, there is a synthesis of mRNA. You know that. Where the mRNA synthesis is happening, just now you told you, it is also happening in the nucleus. But friends, what's the point? The point is that whatever the mRNA synthesized in the nucleus, that mRNA is biologically inactive. It is a immature RNA. We can call it is a functionally inactive mRNA. You can simply call it as native mRNA. It is not active. Friends, if it is not active, how it can undergo the synthesis of protein, how it is involved in the synthesis of protein. So to make it active, we are doing some modifications. 5' capping, you all people know that. 3' polyadenylation, and we will do the mechanism of splicing. What do we call all these processes? Friends, you know, no? All these processes are called as post-transcriptional processes. Is it, friends? Now, where these post-transcriptional processes are happening? Within the nucleus. Once all the modifications are over in the nucleus, then the mRNA become matured. The matured mRNA through the nuclear pore, it entered into the cytoplasm. Not to get confused with that point. Then only, friends, the mRNA can synthesize the protein. Now, the question for the exam point of view, do you think the post-transcriptional processes will happen within the prokaryotic organism? Never ever. All the students all the time remember, post-transcriptional processes are happening only in the eukaryotic organism. Post-transcriptional processes never ever happen in the prokaryotes. What's the reason? The reason is that the process of transcription and the process of translation is highly coupled. Coupled in the sense, friends, both are happening simultaneously in the prokaryotes. Never ever get confused with that particular point. Okay. Finally, friends, protein got synthesized. In the prokaryotes also protein got synthesized. In the eukaryotes also protein got synthesized. Where the protein got synthesized in both organisms, undoubtedly in the cytosol only. And friends, here comes the another interesting point. What was that? The moment protein got synthesized, do you think, do we call that particular protein is a functionally active protein? Never ever. You never call that particular protein is a functional protein. What do you say? You say that the protein is called as native protein. That's a very interesting point, right? Friends, why do you call a protein is the native protein? Such an interesting statement it is. Why do you call a protein is the native protein? See, you take suppose example, we have synthesized the protein called as insulin. How many number of times you heard whenever insulin is synthesized, it never become active. We called it as the proinsulin. Yes or no? Which contain chain A, which contain chain B, and which contain the linker chain, which we can call adapter chain. What we are going to do? We will cut this particular C chain, we'll join the chain A and chain B. Then the proinsulin become mature insulin. So what happened here, friends? The modification happened. Is it? The processing happened. So friends, the same way, I am not only talking about the insulin, every protein that is synthesized in our body, every single protein is called native protein. I'm so sorry, friends. I'm sorry. We call it as the naive protein, not the native protein. Naive means inactive protein. So we have to convert this naive protein into native protein. Native protein means biologically active protein. So if you want to convert this, if you want to convert into active form, again the modification has to occur. Where that modification is happening? Undoubtedly in the cytosol only. And when the modification is happening, is it happening before the translation or after the translation? Undoubtedly after the translation. That's the reason, friends, we are calling it as post-translational modification, not to get confused with this particular statement. So, where the post-translational modifications happens, whether it's a prokaryote or eukaryotes, undoubtedly in the cytosol only. Why? Because protein synthesized in the cytosol only. So I'm summarizing everything now. What I'm summarizing? Let's talk about the prokaryotes. In the prokaryotes, the process of replication, the process of transcription, the process of translation, the process of post-translation, all are exclusively happening only in the cytosol. In case of a prokaryotes, which event is not happening? Which event is not happening? Post-transcriptional processes is not happening in the prokaryotes. Never ever get confused with that point. But friends, let's talk about the eukaryotes now. In the eukaryotes, replication, transcription, and post-transcription. Even I will tell you in the later slides, RNA editing also. All these are all taking place where? All these are all taking place exclusively in the nucleus. Whereas, very important one, the process of translation, the process of post-translation, it is happening where, friends? It is exclusively happening in the cytosol. Friends, never ever get confused with this particular concept. I want you to understand it clearly. Okay. Everyone, please respond. Is it clear so far? Every one of you quickly respond. Is it clear, everyone, so far? Understand where all the events has been taking place. That's a very important. You have to know that. Others. Perfect, perfect. Okay. Now, how to read? See, friends, if you read the molecule, see, in our exam, you can't read anything just basic. If you read very just basic, you never learn the concept. And if you read only basic, once again, you can never answer the question. If you want to crack the exam, your basic should be very nice. From very basic, we have to reach very advanced. Rather than advanced, let's talk about application. Okay, then we will learn the concepts very nicely when we can solve the questions. Now, what we have to do now, anyway, our ultimate goal is what, friends? Synthesizing the protein. But the point is very, very clear. You can't I mean to say, DNA can't synthesize the protein very directly. First, what has to happen? First, replication has to happen, transcription has to happen. So first step of our central dogma is replication. Now, can we enter into the process of DNA replication that much directly? No. You can't study the replication that much directly. In order to understand the process of the DNA replication, everyone has to read the rules that are involved in the DNA replication. And listen, everyone, the enzymes involved in the DNA replication. What, friends? The rules involved in the DNA replication you have to study. The enzymes involved in the DNA replication also you have to study. Then what we will study, friends? Then we will enter into the major thing that is called process. Till that, we have to read rules and enzyme. Now the question is very clear. How can we get the question in the exam? You can get the question directly from the rules. You can get directly from the enzyme. You can get question directly from the process. Otherwise, they will include the rules, they will include the enzyme, they will include the process, and they will raise one single question. So that's the reason, friends, you have to read from very basic, a very complex concept. Okay, now let's talk about first rules. Before understanding the rules, from the 12th standard, we are reading the definition of the DNA replication. Here also, let's read the definition in a one single line. DNA replication. How can I define the word DNA replication? DNA replication is a reaction in which what are synthesizing, friends? Daughter DNAs are synthesizing. From where? From the parental DNA. Is it or not? So you can see the diagram correctly. This is the parental DNA. So this is one parent DNA. This is another parent DNA. Both the parent DNA got separated. The moment they get separated, how do they act like? They act like a template. Template means which is involved in the synthesis of other. So they separated and acting as a template. And now what they are doing, friends? Now they are going to synthesize the daughter strands. Now you have to tell me why. So what's happening here? What's happening? Parent strand is synthesizing the daughter strand. What do you mean by that? Which means the information. What is passing? The information is passing from the parent DNA to the daughter DNA. That's an important statement, friends. That is what actually happening here, not to get confused with that. So transferring the genetic information to the descendant or the next generation with a highest fidelity or with a great accuracy. That's very important, friends. You should not use the word the accuracy should be 99.99%. That is wrong. You should always say that the fidelity or accuracy should be 100%. Even a small information got mismatched also, it leads to huge mutation. That's the reason, friends, the fidelity should be 100%, not it should be 99.99%. Now I have given you a biological definition. Now, if you ask me, sir, please give me the same definition with respect to the chemistry. If you ask me, sir, what exactly the word, remember, friends, what exactly the word DNA synthesis means? DNA synthesis means what? DNA made up of what? Nucleotides, correct, na? So what do you mean by the word synthesis? You are adding one nucleotide with another nucleotide with another with another. That is only called synthesis. That is only called daughter strand. So friends, now you have to ask me, sir, like this, how many nucleotides you can add? Can you count? No, you can't count. See, let's take an example. You take a DNA. You take a DNA and you keep the DNA on the floor. You take the DNA and keep it on the floor. It will move from the planet Earth to the planet Mars and come back. It will back from planet Mars to the planet Earth 600 times. That much long molecule our DNA is. So you can't count how many number of nucleotides are present exactly. That much long molecule it is. So that's why we can say it's made up of billions and billions of nucleotides. So friends, The point is that whenever DNA, I mean to say nucleotides are adding with one another, see how they are getting added. We will discuss DNMP. Why do we call DNMP? Because DNA is containing the nucleotides. We know that deoxyribonucleotides. I will tell you. DNMP represents what? Deoxyribonucleotide monophosphate. N represent what, friends? Number. What is DNTP? Deoxyribonucleotide triphosphate. Friends, such an important statement it is. Whenever one nucleotide is joining with another, it is always joined in the form of triphosphate. Such an important statement it is. It is always joined, what, friends? In the form of a triphosphate only. So DNTP, listen, out of 3 phosphates, only 1 phosphate will be added to this. So it will become N plus 1. And what about the remaining 2 phosphates? They will be released in the form of a PPI. So now this is the first nucleotide, to that second nucleotide is added. Once again, who will come? DNTP will come. From here, once again, only 1 phosphate will come. Then it will become N plus 2. Then another PPI is getting released. In this way, the DNA polymerase enzyme is keep on adding all the nucleotides very fast. We will discuss that in the enzymology. But why I called deoxyribonucleotides? First of all, what does a single nucleotide is made up of? A single nucleotide. Every student commonly know that a single nucleotide is made up of sugar. It is made up of base. It is made up of how many phosphate groups, sir? It is exactly made up of 3 phosphate groups. Sometimes, see, we generally call monophosphate, diphosphate, triphosphate. Sometimes, instead of calling mono, di, and triphosphate, friends, we call alpha phosphate, we call beta phosphate, we call gamma phosphate. Not to get confused with that point. We can call any type of a phosphate, not to get confused, friends. Now look into the picture. So this is called nucleotide, single nucleotide made up of what? Sugar, a base, and phosphate. You know, no, friends, what kind of a sugar is present in the DNA molecule as well as the RNA molecule? We called it as what sugar? Pentose sugar. Correct, na? You know, right? Pentose sugar means how many carbons are there? 5 carbons are there. You all people commonly know that. Pentose sugars are of 2 types. One is called ribose sugar. Another one is called as deoxyribose sugar. Now the question is here, both are 5 carbons only. Then why we are calling one is the ribose and another one is the deoxyribose? Because every student, please look into the diagram. This is the structure of a sugar. Correct, na? This is the ring of the sugar. At the second carbon, if it contain OH group in the second carbon, then only we can call it as the ribose sugar. Why do we call RNA as the ribonucleic acid? Every student remember, the name RNA, the name DNA has given from the sugar only, not from the base or not from the phosphate group. Since it contain OH group in the second carbon, we can call ribose sugar. Suppose if the OH is not present, I mean to say, instead of OH, there is only H is present. Then you tell me, how can I define which is not present? Oxygen is not present, correct? Na? So how can I call it as a deoxyribose? So what is deoxy means? D means no. Oxy means oxygen. No oxygen is present. At the second carbon, only hydrogen is present. That's why I called deoxyribose sugar. So DNA always contain deoxyribose sugar. That's why we can call deoxyribonucleic acid. Such a simple step it is. But this contain 5 carbon. DNA also contain 5 carbon. But there is only lack of oxygen here. Not to get confused with that particular point. Okay, now you understand what is sugar. Now, every student has to understand what are the bases. Every one of you commonly know that there are 2 nitrogenous bases are there. One is called purines, another one is called pyrimidines. All the students all the time remember, all the time, purines are always bicyclic ring, having 2 rings in the structure. Sometimes they can give in the exam like this also, having two rings. That is also correct. Pyramids, friends, always contain a single ring, and it contain what? Only a single ring. Sometimes they will indicate like this also, friends. Here comes the very interesting point. What is that? According to the Chargaff's rule, Chargaff said that the number of purines always, every single time, the number of purines are always equal to the number of pyrimidines. Never ever get confused with that particular point. Purines are always equal to the pyrimidines. If 50 purines are present in the DNA, 50 pyrimidines are present on the another strand of the, I mean to say, in the DNA. And you know very well, purines are adenine, guanine, pyrimidines are thymine, cytosine. Later we will talk about the RNA. According to the Chargaff's rule, adenine pair with thymine with 2 hydrogen bond. You all people know that. Guanine pair with cytosine with 3 hydrogen bonds. Okay. Now you got what are bases. You understand what kind of sugar is present. Now the most important one, very important one. We use it to say DNA is a negatively charged. Why we say DNA is a negatively charged, friends? Undoubtedly because of the presence of the phosphate group only. You have to remember my words very correctly. Without phosphate group, it is impossible to form the phosphodiester bond. You can't form it because if you want to form the phosphodiester bond, you require energy. The energy is coming from what? Phosphate. What do you mean by the word phosphate? Phosphate is the energy-rich molecule. Correct, na? So remember, there are 3 phosphates are there: alpha, beta, gamma, or monoditri. Friends, whether it is the DNA or RNA, the number of phosphates are absolutely same. Okay, I have explained you now. Listen to me correctly. I have explained you what is sugar. I have explained you what is base, and I have explained you what are phosphate groups. But I have explained it individually. But individually, you cannot call it nucleotide, right? If you want to call it as the nucleotide, there is a no doubt, friends. You have to combine them. If you don't combine them, you can't call it as the nucleotide. Now the question is very clear: how you gonna combine? Is there any bonding is involved? Of course, yes. Now all the students listen. This is called a sugar. And the sugar having a first carbon. This is called first carbon. The first carbon of the sugar can be connected to the base, whether it's a purine or whether it's a pyrimidine. How it is connected to the base? With a bond called as what bond? We can call it N-glycosidic bond. Whenever sugar is connecting to the base, I want every student has to remember, when the sugar is connected to the base, we call it as a glycosidic bond. Never ever get confused. So which carbon of the sugar is connecting to the base, friends? First carbon. Now which carbon of the sugar can be connected to the phosphate group, friends? Fifth carbon. So most of the students will think that this is called phosphodiester bond. That is wrong statement. It is not a phosphodiester bond. We called it as ester bond. You can also call it as what? Widely called as phosphoanhydride bond. What do you call? Phosphoanhydride. It is not a diester bond. Never ever get confused with that. So friends, you can see, right? This is called as glycosidic bond. This is called phosphoanhydride bond. But friends, we have discussed about only one nucleotide. But we all people know that the DNA is made up of billions of nucleotides. Then if it wants to form many nucleotides, it is mandatory one nucleotide has to be combined with the another nucleotide compulsory. Otherwise, we cannot synthesize the DNA. How? How is that possible? How they are getting linked? That bond. Listen, everyone. So this is the sugar, right? Sugar contain what groups? Since we are talking about the DNA, right? The second carbon, it contain only hydrogen. What about the third carbon? The third carbon of the DNA always contain OH group, third carbon. And here at the fifth carbon, what groups are present? Phosphate groups are present. Every student listen to me. This is called first nucleotide. This is the first nucleotide. Then I am drawing about another nucleotide also. And so it is also contain sugar. It is also contain a base. It is also contained at the fifth carbon phosphate groups. It also contained at the third carbon OH group. So this is the first nucleotide and this is the second nucleotide, friends. Now listen to me very correctly without confusion. Try to understand. Now the first nucleotide, remember everyone, the first nucleotide at which carbon? Third carbon. It contain what group, friends? OH group. Every one of you listen. The OH group contain what? Oxygen. Oxygen can act as a nucleophile. What do you call? Nucleophile. Why? Friends, oxygen having two lone pair of electrons. You can see that. So since oxygen having a lone pair of electrons, oxygen, listen my words, oxygen nucleophilically come and cut one phosphate group. Only one. Oxygen nucleophilically come and cut one phosphate. Friends, when it cut one phosphate, how many phosphates are left out? Two are left out, right? So they will be released in the form of PPI. Now you can ask me, sir, then how they got joined? Friends, I told you, right? It is cleaving phosphate. Phosphate means what? Energy. So that, understand my words. The oxygen is cleaving phosphate. Phosphate is what? Energy. That energy is used to join the OH with phosphate. This bond, the bond which formed between the two nucleotides, you called it as what bond? Phosphodiester bond. You only look into this. This is the number one nucleotide. Imagine this is the number two nucleotide. So here what group is present? OH group is present. So in the fifth carbon, what groups are present? Phosphate groups are present. So friends, the bond which is formed between the two nucleotides, this bond only we called phosphodiester bond. Never ever get confused with that particular point. Now you just listen. Imagine, friends, this is the first nucleotide, okay? And consider this is the last nucleotide, okay? Now you only look into the picture. This is the first nucleotide. At the fifth carbon, which carbon? Fifth carbon. What group it contain? Phosphate group. Now you tell me, does this phosphate group is reacting with any OH group? No. This phosphate group is not at all reacting with any OH group. And this phosphate group is present at which carbon? Fifth carbon. So we call it as 5 prime. Correct, na? Of course, yes. Now consider this is only last nucleotide. So at the third carbon of the last nucleotide, what group it is present? OH group. You tell me, does this OH group is associated with any phosphate group? No, it is a free because it is the last. So this is called 3 prime. This is called 5 prime. That's why we can call DNA is in the 5 prime to 3 prime direction. And what about the other strand? Other strand is just opposite. It is called 3 prime to 5 prime. That's what we can say. Both strands of the DNA are anti-parallel in nature. Correct, na? Not to get confused with that. So what is the backbone for the DNA molecule? For the DNA molecule, the real backbone is, friends, phosphodiester bond. Never ever get confused with that point. So what we have studied, friends, so far, we studied what is DNA, what definition of the DNA replication. We understand where the transcription, translation, replication, all the processes are happening. Now we have to study the process of DNA replication very elaboratively. If we want to read the replication, it is mandatory. We have to understand the rules. We have to understand the enzymes. After understanding these two, the process become very easy for us. So that's why we have to focus on the rules and enzymes. So let's talk about it. What are the rules? Semi-conservative. Starts at the origin. We will discuss one by one. Synthesis always. Synthesis means the daughter strand. It should always synthesize in the 5 prime to 3 prime direction. The direction of the DNA replication may be uni or bi. I'll explain everything briefly. Semi-discontinuous RNA primers are required, high fidelity or accuracy. Now, you all people has to ask me one very good doubt. Sir, is it necessary that the DNA has to follow every single rule? Can it skip any rule? Remember my words. The DNA fail. If the DNA fails to follow even a single rule also, if it fails, the DNA never start the process of replication. Suppose example, you're writing a CSIR, you're writing a GATE, or you're writing IIT JAM exam. If you are not meeting the eligibility criteria, you are not eligible to write the exam. So even though you're not meeting the eligibility criteria, if you try to write the exam, so no, the application process only won't allow you. Similarly, if the DNA is trying to escape or trying to skip these rules, the replication never starts only. If the DNA wants to undergo the replication, it is necessary or mandatory. It has to follow every single rule. It has to pass every single rule compulsory. So now let's talk about one by one rule little elaboratively. So the first rule is called semi-conservative. You read many times this rule, but let me explain a little elaboratively here. Out of 12 question papers, minimum 6 to 8 times the question was appeared from this particular experiment. So that's why it is very, very important. I'm requesting all of you to understand this experiment very briefly and very, very elaboratively. Now you have to ask me, sir, what is the semi-conservative? How do you have given the name? Remember, I told you, right? The Watson and Crick decided. What they decided? They have to propose a model. I want you to understand me, friends. They have to propose a model. By using that model, the DNA can undergo the replication. So what model Watson and Crick proposed? Semi-conservative model. You know what is semi-conservative model? Watson and Crick said that according to the semi-conservative model, the two parent strands get separated. You can see that. The two parent strands get separated and the two parent strands are going to give rise to what strand, friends? They are going to give rise to the daughter strand. Such a simple model they proposed. Two parent strands are separating. There is a synthesis of the daughter strand. But friends, the point is that unfortunately, see, who proposed semi-conservative model? Whomever discovered the DNA, structure of the DNA, Watson and Crick. Most of the scientists believed their model, most of the scientists. But unfortunately, what is the proof? There is no structural proof. There is no evidence to prove. What to prove? The DNA always follow the semi-conservative model only during the DNA replication. There is no proof, right? So by opposing the semi-conservative model, some other scientists proposed some other models. Conservative model, dispersive model. According to the conservative model, very simple point it is. Entire parent DNA should be preserved and giving rise to the daughter DNA. What? Entire parent DNA should be preserved and giving rise to the daughter DNA. And what will be the third model called as dispersive model? I can call it cut and patch model. Like small parent, daughter, parent, daughter, parent, daughter, parent, daughter, parent, daughter, like this. So friends, how many models are proposed total? 3 models are proposed. Most of the scientists accepted which model? Semi-conservative model, because it is proposed by Watson and Crick. But there is no proof. So to prove this, what to prove this? To prove DNA always follow the semi-conservative model only during the process of DNA replication. It never follow conservative model. It never follow dispersive model. To prove this, semi-conservative model only idealized model. Semi-conservative model only correct model during the DNA replication. The two intelligent scientists, Matthew Messelson and Franklin Stahl, they have done classical experiment. We can say that till today I'm talking, till today in the field of life sciences, in the field of biology, this is one of the most classical experiment that ever done. Very simply, they have proved the DNA always follow semi-conservative mode only. Okay, how they have proved, we will discuss in the tomorrow morning class. Okay, is it clear everyone now? Let me know if you have any kind of a doubt. I will re-explain. Every one of you. And friends, how to read now, that is very important. So let me explain you 2 to 3 classes. After 2 to 3 classes, I will explain to everyone how to read a point by point, how to read step by step. I will explain you nicely, then you will just follow it. I hope everyone understand the concept very clearly. I am telling you once again, friends, your exam depends on how depth you learn the concept. The more depth you learn, undoubtedly you are going to crack the exam, okay, with good practice. Okay, in the evening we are having 6:30. I told you, right, today and tomorrow also we are having all classes in the Zoom only. From the Wednesday onwards, we are having all the classes in the app only. Okay. So evening 6:30 p.m. we will share the link. I will start the human physiology chapter and I will give you the timetable also in the evening class. Okay. Okay, friends, I'll share the PPT to you, all of you. Okay. Thank you, all of you. I'll see you in the evening sharp 6:30 p.m. Okay. Thank you, all of you. Thank you so much.